Hello my dear students, welcome back to my channel. So now after a long time I started. So now I want to discuss about viruses. In this series of videos I will be discussing all about viruses. So we know these days we are very well affected, or we are either getting affected or getting affected or indirectly getting affected with coronavirus. So I found a need to just discuss about what are viruses, all about all aspects of viruses, and I will try to give you towards the end of my session, I will try to give you some information about coronavirus also. So first in few videos I'll try to give you all the basic aspects of viruses. So today now I'm going to start with the first, introduction about viruses. So viruses, they form a unique group of biological entities. Unique group of biological entities, and they show both living and non-living. You know, viruses will be both living and non-living, they show. And they are acellular organisms or non-cellular. Why we call them as acellular or non-cellular? Because they do not show proper cell structure. There will not be any cell structure. Viruses have only two components, that is nucleic acid and the protein. Nucleic acid will be present towards the center, that is forms a core, and protein will be present towards the outside. Protein is called as a capsid. That's why viruses are also called as, nucleocapsids. They are called as a nucleocapsids. Viruses are also called nucleocapsids. Now, viruses are considered to be the connecting link between living and non-living. So, let us see what are the living and non-living characters of viruses. So, living characters of viruses are, they have DNA or RNA. No virus will have both DNA and RNA. It will have either DNA or RNA as genetic material. And it can multiply inside the host. The viruses can multiply only inside the host, and they can undergo mutation. Since either DNA or RNA is present, they can undergo mutation. Now, what are the non-living characters? They are not composed of cells. Just now I told you they are acellular or non-cellular. That means they do not have proper cell structure. They cannot be observed under light microscope. Very, very small they are. And then they are crystalline outside the host. They are crystalline outside the host. So, viruses are considered to be obligate intracellular parasite. Obligate intracellular parasites. They are considered to be obligate intracellular parasites. And the study of viruses is called as virology. Study of viruses is called as virology. That is about little bit of introduction. The next I am going to discuss is what are the discovery made in this field. What are the historical aspects and what is the discovery made. For the first time, the word virus or as venom or as poisonous fluid was given by Pasteur. So Pasteur called these viruses as venom, or the term virus was coined, and it was considered to be a poisonous fluid. Considered to be a poisonous fluid. The next other scientist, Ivanovsky, Russian scientist Ivanovsky was the first one to observe the virus tobacco mosaic virus. He was the first one to study tobacco mosaic virus, and he studied the infection, that it is infecting the host plant, that is tobacco plant. And when this extract from the virus extract was taken from the plant, it was observed that these organisms were passing through bacterial filters. They were easily passing through bacterial filters. They were easily passing through bacterial filters. So therefore, these were called as filterable agents. They were called as filterable agents. So the word filterable agents was given by Ivanovsky. It was given by Ivanovsky. So that is about the discovery. Okay, next one. Now, after Ivanovsky, the next study was done by Weizsäcker. The scientist, next one was given by the scientist Weizsäcker. So Weizsäcker called the viruses as contagium vivum fluidum. Contagium vivum fluidum. It means it is a living infectious fluid. Contagium vivum fluidum means it is a living infectious fluid. He tried to apply the extract from the diseased plant to the healthy plant. And he found that the healthy plant caused disease in healthy plants also. And thus he thought they are the living infectious fluid and called them as, in Latin, called them as contagium vivum fluidum. Filterable agents was called by Ivanovsky, contagium vivum fluidum by Weizsäcker. Then the other scientist who studied, W.M. Stanley. Other scientists who studied are W.M. Stanley. He was the first one who performed crystallization of tobacco mosaic virus. He was the first one to crystallize this tobacco mosaic virus. And then the other scientist, Frankel Conrad. Other scientist Frankel Conrad was the one to find out what is the genetic material in tobacco mosaic virus. And he observed that it is the RNA which is the genetic material in case of tobacco mosaic virus. It is the RNA which is the genetic material. And he used several techniques. The three important techniques which he used to understand viruses are, one is called as ultracentrifugation. One is called as ultracentrifugation, and then electron microscopic technique, and the other one is X-ray crystallographic technique. So these three techniques were used in order to understand viruses. What is the use of ultracentrifugation? It helps in separation from other components, or viruses can be separated based on their size or based on the structure. And X-ray crystallography helps in understanding the molecular structure. Similarly, electron microscope also helps to observe the structure at a molecular level. And what exactly is the molecular structure can be identified by X-ray crystallographic studies. So, I have made a only very short video, children. So, this is just I discussed starting with introduction and then a little bit about discovery. So, the my next video, I am going to discuss about the structure, composition of viruses and symmetry of viruses. So, I wanted to discuss in part by part instead of discussing completely in 1 hour video. 1 hour video will be too lengthy, it will be difficult to watch, many will not. So, what I thought for best purpose or best understanding purpose, I am going to just stop it here. Then my next video, I'll make it little more lengthier in my next video. I will try to discuss about the structure of viruses, composition, their symmetry, types of viruses, and then multiplication of viruses. So these we will relate along with the transduction. So that I'll make it at a stretch, because once you understand the structure, all the other things should be done at a stretch. Okay, students and all other my viewers, my subscribers, Thank you so much for listening. Please share to the maximum extent and ask all your nears and dears, all your students and other beneficiaries, those who want to get benefit, you ask them to subscribe. Thank you very much. Have a good day.